Today we're going to be talking about bacterial infections, and we're going to cover TB, pneumococcus, and Staph aureus, and so three very, very different pathogens. And hopefully you'll see I've tried to organize these in a way that'll sort of bridge the types of intracellular pathogens we've been talking about thus far in terms of the different acute and chronic viral infections and into a really different kind of problem for the immune system, which are these extracellular pathogens that can't be targeted in the same way as an intracellular virus. So we'll start with TB. So bacteria obviously are divided into different groups based on their genus and species and larger subtypes. And so TB is an acid-fast rod-shaped bacillus, and its acid-fast is the description that was used sort of functionally to stain it back before there were genetics. And the reason it's acid-fast is that it has this unique wax-rich cell wall composed of long-chain fatty acids and glycolipids. So it's just like sitting in a globule of fat, and that actually provides it with a lot of protection from immune surveillance. It has 250 genes dedicated to fatty acid metabolism and a very slow 20-hour replication time. So it has quite a big genome, so 4 million base pairs. We were talking about yesterday about SARS-CoV-2, and I was saying SARS-CoV-2 was a really large virus and it had 30,000 base pairs. And then herpes viruses were, I said, even larger, and there we're talking in the hundreds of thousands of base pairs, and now we're up to the millions of base pairs. MTB is widely distributed throughout the world with obvious concentrations in certain areas where it's a major public health problem. Causes 2.2 million deaths per year, has a humongous burden of disease when measured as disability life years, so 45 million total dailies. 2 billion individuals infected with MTB, so a third to a quarter of the world. 10% have a risk of developing disease following infection. So some people do control MTB, and untreated disease mortality is 50%. 8 million new tuberculosis cases per year, 1 new case every 4 seconds, and 10 to 15 individuals infected annually by a single untreated patient. So the R naught for this is quite high. MTV is, you can almost think of it a little bit like HIV in terms of analogy, because it actually uses the cell that should kill it as its primary host. And so I think, again, thematically, these really, really successful pathogens are the ones that have somehow co-opted critical effector mechanisms of the immune system in their life cycle. And so what you can imagine up here, this line here denotes an airway, so the alveolar sac of your lung. And in each alveolus, you have usually an alveolar macrophage that just sits and hangs out and eats the dust and things that you breathe in. And so it's a happy little macrophage sitting on the corner of your alveolus, and mycobacteria comes in and is eaten by the macrophage. And the macrophage immediately knows that this is a pathogen, and so it's going to try to initiate an immune response along the lines of what we talked about. It will secrete cytokines, and it will actually traffic into the blood vessels of the lung to try to recruit other cells to come deal with the infection. And so that will actually bring other macrophages in that can then be infected by TB. And the immune system starts to try to wall these macrophages off and control the infection. And they'll form a ring of lymphocytes, of T cells around them, and we'll talk in more detail about what those T cells are, and rings of other immune cells. And this structure actually becomes incredibly organized and regulated by chemokine signaling and other receptors, and eventually walls itself off physically by secreting a fibrous cuff around the center. And this is happening because the bacterial infection is not being cleared, and we'll talk about why that's happening as well too. But this whole structure eventually that forms, we call a granuloma. And this can be incredibly stable for a long period of time, and it can actually protect the host from the spread, the dissemination of the bacteria. But at some point, sometimes years after the formation of these granulomas, the bacteria reactivate and start growing and killing all of the cells inside of this granuloma, and eventually breaking out, forcing themselves back into the airway, at which point you cough and you spread this to the people around you. So this is what this looks like pathologically. You can see these granulomas forming here in the airways. So you have these nice airways, and then this blockage formed by these granulomas. And you see this lung that's just becoming totally fibrotic. It should be sort of spongy and squishy and pink, and it's just becoming bloody and these fibric patches here. And again, so you get this really super well-organized structure that does a good job, again, for years of controlling the bacteria, but you get this slow dissemination throughout the lung. Again, kind of thinking of it like HIV, where there's control, but there's this also slow degradation of capacity, and eventually you get these lesions that break through and cause the disseminated disease and destruction of the lung and destruction of and spread to other people. So MTB is recognized by those innate receptors in the macrophage immediately that we've talked about. And so these include the TLRs that recognize actually some of those lipid ligands from MTB, and then also some of the NLRs inside the cell that are going to recognize and mount an inflammatory response. And so they'll trigger a wide range of inflammatory responses, including something in TB that's called the cytokine storm. The cytokine storm, unfortunately, has become, I think because people like the phrase, overused. It got massively overused during COVID. And it doesn't really have a strict definition, but in pre-COVID days, it was usually restricted to cases where there was a feed-forward, out-of-control amplification of inflammatory signaling driven by cytokines. Now it kind of means any level of cytokines that we think is too high, and we're going to call it a cytokine storm. But there is this positive feedback loop in TB that's really critical to the formation of disease, where macrophages initially respond, even if they fail to clear. They recruit other macrophages and monocytes, inflammatory cells to the forming granuloma, which then secrete more cytokines and recruit more cells to form the granuloma. And then eventually recruits the traditional adaptive immune response, which will try to clear the infection and drive this classic type 1 intracellular targeted pro-inflammatory response to get the macrophages to kill the bacteria. So why do the bacteria persist? So the way that these bacteria are going to be killed, because they're living inside the macrophage, they're brought in by phagocytosis into an endosome, just like our viruses. And I mentioned that our macrophages like to bring things in that way, and then they try to kill anything that's in those endosomes by acidifying them. So they will start to acidify them and create sort of a metabolically unpleasant environment for everything that is in the endosome by fusing with different lysosomes. These can get incredibly low pH, pH 4.5, even lower. If this works, if the macrophage could actually get through this entire cycle, it would actually kill the TB. And so this is what we're really trying to drive to, because if the pH gets low enough, it's actually a unhappy environment for the TB. And so you have this central role of the macrophage here. You've got these bacterium living in this endosome, and you've got the TB trying to block the endosome from maturing and getting acidified. So the cytokine storm that's initiated by the innate response will determine the character of the ensuing adaptive response. There's roles for lots of different cell types here, including traditional alpha beta T cells, which we're going to be talking about, CD4 T cells in particular, but other unconventional cells that we haven't spent a lot of time on but including NK T cells, gamma delta T cells, these cells that sort of are lymphocytes, but that sit at the border of innate and adaptive immunity. And the function of all of these lymphocyte populations is really critical for the macrophage to be able to maintain its control of the bacteria. And so just as a reminder, we sort of broadly characterize the immune response into these large types of responses. So we have the type 1 response, which we've almost exclusively talked about for intracellular pathogens, the type 2 response, which controls anti-helminth worm responses and is also associated with allergy, and the type 3 or type 17 response, which we associate with extracellular bacteria, and we're going to talk about in the context of staph and strep here in a minute. And then against those responses, we've got the role of regulatory T cells, which again, we've touched on just a little bit, but are a differentiation state of CD4 T cells that can occur while the T cell's developing in the thymus and can also occur in the periphery. And the goal here is for these cells to shut down pathogenic T cell responses that are causing too much tissue damage. So there's always a negative cycle, a negative feedback loop somewhere in the immune system to try to turn things off if it gets out of hand. And so we're going to have this response trying to drive the response up and response trying to push it back down. And this is really probably what controls the diversity of outcomes we see with MTB. So remember, most people that get infected with MTB actually do okay for a very long period of time. But so many people are infected with MTB that the relatively significant percentage of people that don't do okay, certainly a much larger percentage of people that don't do okay than, say, with COVID, make it an incredibly high burden disease in terms of pathology and poor outcomes. And so it's this balance of regulatory versus effector signals and the various types of those signals that determine the activation milieu of the granuloma and the infected macrophage. So if you've got great Th1 cells making interferon gamma and K cells making interferon gamma, other innate inflammatory cells making TNF and interferon gamma, they're going to tell that macrophage to just really start killing that bacteria, acidify those endosomes, make reactive oxygen intermediates, reactive nitrogen intermediates, really be as inflammatory as you can be to kill that bacteria, which is what you're going to have to do because those bacteria are super, super strong and resistant to death because they've got that big waxy coat around them. It's like encased in a candle. If you've got the wrong kind of signal coming to that macrophage that says, this is less of a problem, it's not an immediate threat, it's more, maybe it's like a worm. That's why if you have a Th2 cell, maybe you can try to tolerate for a while. It's more of a wound, then you're not going to kill the bacteria. And if you've gotten too inflammatory up here and your genetics and your immunological history have promoted this more regulatory response and secreting of cytokines like IL-10, which are suppressive cytokines, you're also going to shut down the antimicrobial response of the macrophage. And so it's going to be this war between these two types of signals, both of which have sort of a good purpose you can think of. One is this pro-inflammatory signal to kill the bacteria. The other is an anti-inflammatory signal to make sure you don't tear your lungs apart in trying to kill the bacteria. And in terms of, and particularly you'll see this in a lot of models, and again, it's modeled because it's something we can measure easily. The main cytokine that sort of suppresses pro-inflammatory responses is IL-10, interleukin-10. So it's a key regulator of pathology in multiple infections. In flu, for instance, IL-10 is produced by multiple cell types, and it's required for survival in certain models because you also don't want your immune response running out of control. And the pleiotropic effects of the cytokine are actually still really poorly understood at a mechanistic level. IL-10 can work on almost every cell type, and it will cause it to stop doing what it's doing really, really quickly. And we don't really understand all the signaling pathways that it uses to do this. It's incredibly, incredibly potent. And so it can be made by many, many different cells and act on many, many different cells to shut down the response. So This is sort of a summary of how all of these pathways are going to work to try to actually control and kill TB. So you've got all of these lymphocyte populations that, regardless of how they get there and how they get activated and what specific antigen they're seeing, their main role in this type 1 intracellular response is to make lots of interferon gamma to signal to the macrophage. And it's because the macrophage has to be the cell that makes the decision to kill TB. So this is different than in flu or in SARS-CoV-2, where you have an epithelial cell, or even in these herpes virus infections where you have B cells and T cells that are infected. Here, the macrophage is going to kill the bacteria, and it's going to kill it by metabolically destroying the bacteria with acids, with reactive oxygen intermediates, and it's going to have to super inflame itself and dump all of these really toxic chemicals inside the phagolysosome and kill the bacteria. It also will try to starve the bacteria of essential nutrients that it needs. And if that fails, then you've got this long-lived granuloma that can eventually sort of almost stochastically break apart and release the bacteria. And so one theme you should see here is interferon gamma, interferon gamma, interferon gamma. It's this type 1 response that we're trying to make. And so if you look in animal models and just ask, well, what different pathways are most essential for control of TB in animal models? And the mouse model for TB is not so bad. It's I'll point out later in the talk today where the mouse model is not good for certain things. But for TB, it's generally pretty good. And a common way in immunology that we assess the significance of a particular pathway for a particular pathogen is just by knocking out that particular gene. And so if you knock out interferon gamma and you look at MTB growth, it's basically unrestricted. So if there's no interferon gamma, this is the worst possible outcome for a mouse. Interferon gamma is the essential thing. If you go to the other end of the spectrum, in a wild type animal, this is sort of the level of control, the set point of TB that you get. So you get this initial exponential growth, and then over time, over a year, you get this sort of flat line. Again, just sort of like HIV, control in the lung of this détente between the immune system and the bacteria. And that's even true if you get rid of gamma delta T cells, which are one of these innate cell types. There's enough of the other lymphocytes making interferon gamma that you don't really need them. If you get rid of class 1, what that does in effect is it gets rid of all your CD8 T cells, because your CD8 T cells can only see target cells through class 1, that class 1 MHC molecule. So this is getting rid of any way for a CD8 T cell to see a target cell, and you lose a little bit of your control, but not very much. So CD8 T cells, even though this is an intracellular pathogen, are not really important because they're not actually killing the macrophage to get rid of the bacteria. They're just secreting interferon gamma to tell the macrophage to kill the bacteria itself. Because if they just tried to kill the macrophage, it wouldn't actually kill the bacteria, because the bacteria would probably be safe inside the endosome, and as the macrophage fell apart, the bacteria would just escape. If you get rid of class 2, this is MHC class 2, that's going to restrict the ability of CD4 T cells to see antigen and to be activated. So you basically don't have CD4 T cells in this animal. And now you're really losing control of the bacteria. There's a little bit of control, but there's, it's still basically exponential growth of the bacteria. So CD4 T cells are absolutely essential, and it's because they're going to be making the largest amounts of interferon gamma, the largest source of interferon gamma, to tell a macrophage to kill the bacteria. If you get rid of all alpha beta T cells, so that's CD4s and CD8s, it's even worse. So you're sort of adding these two losses here. If you get rid of NOS2, so this is this enzyme that's going to make these reactive nitrogen and reactive oxygen intermediates, that's even closest to interferon gamma loss. So that's going to be the effector enzyme inside the macrophage that's making these really toxic chemicals like peroxides that you're going to dump on these bacteria that will finally break through that waxy coat and kill them. And so it's this combined adaptive innate response working together. It's this adaptive response T cells telling this innate effector to use every weapon in its armamentarium to kill the bacteria. So that's genetic deficiencies in mice that we engineer to prove significance. There are a few labs that work on humans to try to identify genetic deficiencies or genetic variations that reveal susceptibilities to certain pathogens. And so if we look at these human genetic deficiencies that are associated with very, very poor outcomes to Mycobacterium broadly, which are a very big family of bacterium. There's many different species of mycobacteria that infect different animals. So like there's Mycobacterium avium, Mycobacterium bovis, so cows and birds, and we happen to get Mycobacterium TB. We find that people that have natural mutations in the interferon gamma receptors, in IL-12, which is a key cytokine that promotes interferon gamma production, and then the signaling molecules downstream of interferon gamma are particularly susceptible to Mycobacterium. So the primary phenotypes of individuals with genetic deficiencies in interferon gamma signaling or activation is susceptibility to mycobacterial disease. It's usually actually not TB that they present with, because they'll present very young. And they'll usually present with Mycobacterium avium or Mycobacterium bovis as a skin infection. Also Mycobacterium leprosy. So leprosy is also a mycobacterial disease. And a lot of these mycobacterium are just in our environment all the time. They're in the dirt, they're in the, if you're ever around animals, you'll be exposed to them, but very small amounts of interferon gamma and macrophage activation are enough to clear them from an otherwise healthy individual, unless you're missing these pathways. And this is really in contrast to type 1 interferon deficiencies, which are manifested usually as very early, rapid susceptibility to viral infections. I'm happy to talk more about that. That's sort of a growing area of research now that genetic sequencing has gotten much cheaper, is to use people as sort of model knockout animals and say, well, we're just going to look at people and see what they get naturally and see what they struggle with. And then we're going to sequence them and see if we can associate any of their genetic variation with their particular health profiles. So summarizing so far what I've shown you for Mycobacterium, MTB is never completely cleared following initial infection. It's chronic. It lives in your macrophages for the rest of your life. The primary effector mechanisms are macrophage bactericidal functions, and their success is determined by the cytokine and cellular regulatory environment. And really small, subtle shifts over time or dramatic short-term changes lead to reactivation of disease. So the small, subtle shifts is something closer to HIV, where there's this constant, intense battle between your immune system and the mycobacterium, but it's happening at a very low level in these granuloma structures in the lungs. But eventually, in a significant number of people, the mycobacterium win, versus in if you have a short-term event, some sort of massive immunodeficiency, you get HIV and it progresses and you lose your CD4 T cells that are going to be so critical for making that interferon gamma, or you have an immune suppressive event because you go on steroids for some reason for some other auto-inflammatory condition. All of these will then massively reactivate your TB. You'll also see people, like if you watch TV and you see commercials for these anti-inflammatory drugs for autoimmune conditions, they'll often say, if you have TB, you can't take this because there's this ongoing war with your immune system that you really can't disrupt. A lot of those are TNF blockades. And so this figure really summarizes everything I've just told you, that you've got the macrophage as the host of the bacteria, and then you bring in the adaptive immune response to continue instructing that macrophage to try to contain the bacteria and destroy the bacteria and not let it grow and not let it burst out and spread.